Welcome to the virtual roadshow series presented by Channelchek and Noble Capital Markets. Of the 6,000 small and microcap companies listed on Channelchek, today we're featuring Genprex, NASDAQ ticker symbol GNPX. Research coverage of Genprex is provided by Noble Capital Markets. Noble is a FINRA-licensed, SEC-registered broker-dealer. All Noble research is available on Channelchek. Following the formal presentation, Ahu Demir, Noble's biotechnology research analyst covering Genprex, will ask our presenter a selection of the questions received. The source of the questions will remain anonymous. And now, here is Rodney Varner, CEO of Genprex, to make the presentation. Rodney, take it away.
Okay, thanks very much. Thanks to Noble for giving me the opportunity to present today. I'm Rodney Varner, a founder, chairman, and CEO of Genprex. At Genprex, we're reprogramming the course of cancer and diabetes through gene therapies. I know that this audience has seen the forward-looking statement slide many times. I commend it to your reading. This deck is available on our webpage, and you can read it in full there if you would like. This is our basic stock information at a glance. This information was as of August 28. You can see we trade under GNPX on NASDAQ. Our share price on that date was $3.88. Market cap 150 million. 10-day average volume 1.43 million. We have about 38 million shares outstanding. As of June 30, we had cash of 22 million on the books and no debt except for working capital bills in the ordinary course of business. I want to spend some time on this slide. I know that in these sessions, many times people are not able to listen to the entire session, not able to focus on all the slides that are given. But these are the high points that I think potential investors should consider. First of all, we have a gene therapy platform. We can deliver multiple genes. We have in-licensed from MD Anderson a series of tumor suppressor genes, all to fight cancer. We can deliver these genes to multiple types of cancer, and there's preclinical data from MD Anderson and other institutions that indicate that our lead gene therapy may be effective in a wide range of cancers. We can also combine our gene therapy with a number of different cancer-fighting drugs, including the largest selling drugs in lung cancer, Keytruda and Tagrisso. We are soon going to be initiating 2 clinical trials combining our gene therapy with the top-selling lung cancer drugs, Keytruda and Tagrisso. We have a fast-track designation for use of our gene therapy in combination with Tagrisso in patients whose disease progresses after they begin taking Tagrisso. That's about 100% of Tagrisso patients. Nearly all of them progress after they begin Tagrisso. According to AstraZeneca's own data, that median time to progression is between 14 and 18 months. So we're positioned to begin treating these patients with our gene therapy as they progress on Tagrisso alone, which puts us in a position to treat many, if not most, of the patients who come off of Tagrisso or who would otherwise come off of Tagrisso. When combined with our gene therapy, they'll just continue on Tagrisso in combination with our gene therapy. Tagrisso sales again are about $3 billion, or were in 2019. We have demonstrated clinical achievement. We've done 2 phase 1 clinical trials with our gene therapy, one of them as a monotherapy, one in combination with the EGFR inhibitor Tarceva. Like Tagrisso, Tarceva is an EGFR inhibitor. It's I view it as an earlier generation of the EGFR inhibitor drugs like Tagrisso. In its day, Tarceva was the largest selling EGFR inhibitor in the United States. Tagrisso has taken over that designation and is now prescribed for more EGFR positive patients than other EGFR inhibitors. We're initiating these two trials in early 2021. One of them is going to be with Tagrisso, the other with Keytruda. Keytruda is Merck's largest selling drug with sales of about $9 billion in 2019. With the combination, or with Keytruda and Tagrisso, we're covering a large majority of lung cancer patients. So that majority of lung cancer patients will be eligible for combinations with our gene therapy and Tagrisso or Keytruda. We have world-class academic partners. Our oncology technology all comes from MD Anderson Cancer Center in Houston, where we've had a 10-year relationship with ongoing preclinical research there at this time. We have an expanding pipeline. We're expanding our oncology platform with our preclinical program. Researchers at MD Anderson are identifying new drug combinations and additional cancers to be targeted all along, and are continuing to work on that now. Our diabetes therapy comes from the University of Pittsburgh. We have a strong balance sheet. As of June 30, we had about $22 million in the bank and no debt. Now, I want to spend a few minutes more on the Fast Track designation, what that is and what it means to us. We received the Fast Track designation earlier this year. It's for the combination of our gene therapy with Tagrisso, which had about $3 billion in gross sales in 2019. A Fast Track designation can be awarded for therapies that show the potential to address unmet medical need for a serious or life-threatening disease. The therapy has to show a potential for improvement over existing therapies in the targeted population. In our case, the FDA found that our gene therapy, when combined with Tagrisso, has the potential to be an improvement over existing therapies in that patient population. We think that's a big deal. We think that's validation of our program and the potential of our gene therapy to treat a large number of lung cancer patients. The Fast Track designation helps facilitate drug development. It expedites regulatory review and opens doorways for accelerated approval and other procedures that might ease the pathway to drug approval. So all in all, the Fast Track designation was and is a major accomplishment for us, and we're really proud to have that validation. Let me touch on our pipeline. We have, again, we did a phase 1 clinical trial using our gene therapy alone in late-stage lung cancer. I'll show you that data here shortly. We did a phase 1 and a portion of a phase 2 using our gene therapy in combination with Tarceva, the EGFR inhibitor. And it was the data from those two trials, along with preclinical data, that led to our receipt of a Fast Track designation from the FDA for combining our drug with Tagrisso or osimertinib in late-stage lung cancer. Now, interestingly, erlotinib or Tarceva and osimertinib or Tagrisso are both EGFR inhibitors, but they're different drugs. Nevertheless, the FDA allowed us to continue our program using osimertinib, even though our phase 1/2 trial was with Tagrisso. We assumed that the FDA considered their mechanisms of action to be similar enough to allow us to essentially switch drugs in the course of the fast track application and receive the fast track for osimertinib or Tagrisso. That's really important because Tagrisso has become the largest selling EGFR inhibitor in the US by far, and its sales are growing rapidly. So that switch was a really positive thing for Genprex. Next, we have the combination of our gene therapy with Pembro or pembrolizumab, also known as Keytruda. We have extensive preclinical data from MD Anderson suggesting that's going to be an effective combination, and we're planning to start that clinical trial early next year. We also have the preclinical program of a gene therapy for treatment of diabetes that was developed at the University of Pittsburgh. We in-licensed that earlier this year. Importantly, the University of Pittsburgh just a few months ago received a $2.6 million NIH grant to support primate studies of this gene therapy. The studies were already going on at the University of Pittsburgh. This funding will fund additional primate work there and hopefully lead to an IND for clinical trials of this program. We think that NIH grant is very important for a number of reasons. First, it's again validation. Those grants are peer reviewed, and 2.6 million is a large grant from the NIH. So we feel that that peer review and NIH grant is important validation of the technology. And then also this will fund additional primate research at the University of Pittsburgh that either might not have been done or that we would have had to pay for. So a very positive thing for Genprex. Now I'll address the non-small cell lung cancer market. Our lead program again is in non-small cell lung cancer. Lung cancer is the second most common form of cancer and kills more people worldwide than any other form of cancer. It's one of the largest, if not the largest, cancer market. Our initial target is non-small cell lung cancer, which constitutes about 84% of lung cancers. So the largest subcategory of lung cancer. There are about 2 million cases a year worldwide, 228,000 a year in the US. Again, we're combining with Tagrisso and Keytruda. Tagrisso's sales over $3 billion in 2019. Keytruda's over $11 billion in 2019. And this lung cancer market is expected to grow to $26 billion by 2023. So very large markets, serious unmet needs. Now, I want to zero in a bit on the potential for our gene therapy when combined with these other classes of therapy and the magnitude of the unmet need in late-stage lung cancer. If you look at the slide before you, the column targeted therapies, you'll see only about 10% of lung cancer patients are even eligible to receive a targeted therapy. That's like Tagrisso. The rest of the patients don't even receive that type of drug. Even so, even though the market is only 10% of the patients, Tagrisso has had these very large sales. And unfortunately, even for the 10% who do receive those drugs, the drug resistance becomes inevitable over time. These patients relapse. And so there's a serious need for additional drugs after these patients relapse. At this time, these patients after relapse will normally receive a chemotherapy, most likely. And so this is the population that the FDA agreed was a serious unmet need that our drug may address. As the patients come off of Tagrisso, or would come off of Tagrisso, but for our drug, we can add our drug to Tagrisso and continue them on that treatment so that we can extend the time that they can benefit from Tagrisso. There's a similar situation with immunotherapies. In the case of immunotherapies, only around 12% of patients are eligible and benefit from immunotherapies. That's a slightly different definition than with the targeted therapies. This 12% is patients that are eligible for and benefit. And again, few patients obtain any durable benefit, and many relapse with drug-resistant disease. So these patients, again, they receive an immunotherapy. Many of them do very well for quite some time, but ultimately, most of them relapse. Again, our initial approach is to target these patients who relapse on an immunotherapy. And basically, we can be at the end of the funnel as these patients come through. They're treated with an immunotherapy, but then need something else. And then additionally, in addition to those two prospects, preclinical data also indicates that our gene therapy can expand the number of patients that can benefit from these therapies. That is, make that 10% and 12% larger, more people eligible to begin with. So that if you consider that, you can see that the potential is really vast. But we will be quite happy to receive approval to treat patients who become resistant to targeted therapies or immunotherapies. Those are both really giant markets. So this slide illustrates our platform. I mentioned we have multiple tumor suppressor genes in-licensed from MD Anderson Cancer Center. There's a whole series of them that can be delivered with our nanoparticle delivery platform. We've shown multiple drug synergies. Researchers at MD Anderson have shown that our gene therapy is synergistic with a range of both targeted therapies like Tagrisso and immunotherapies like Keytruda. And then finally, they've shown that our gene therapy can be beneficial in a wide range of cancers as well. And also I might add that our delivery platform has the potential to deliver a wide range of genes in addition to those that we've licensed from MD Anderson. It's not limited to those at all. And we may very well be pursuing treatment of cancer or other diseases with genes in addition to those that we've already licensed. So let me talk a few minutes about that platform. The delivery platform is very important. Most gene therapies use a viral delivery system. In fact, our diabetes delivery system is a viral system. That's the one developed at the University of Pittsburgh. We don't use that in cancer. We use this non-viral nanoparticle platform. Dr. Roth, our lead inventor from MD Anderson, is a very experienced, more than 40-year researcher and lung cancer surgeon from MD Anderson. And he was one of the early movers in gene therapy in lung cancer and other cancers. He used viral delivery systems back in the 1990s and did extensive research with them. In fact, conducted several clinical trials using viral delivery systems. But he identified, well, along with many others, limitations to viral delivery systems. They sometimes generate immune responses, limiting the number of times they can be used in a patient, they have other potential side effects that are not favorable for patients, and they don't target tumors, and particularly lung tumors. And so what Dr. Roth wanted to find was a less toxic system that could be delivered systemically, that would circulate through the blood system and attach to tumors wherever they're found. And therefore, he developed this system, our nanoparticle delivery system. It uses a positively charged lipid nanovesicle to carry plasmid DNA to tumors. It has a positive electronic charge, and it's attracted to cancer cells. It's been shown in biopsies that there's a 10 to 25-fold uptake of these nanoparticles in cancer cells versus normal cells. And importantly, this is not just theoretical. We've treated over 50 patients with this delivery system and shown that it has a very favorable safety profile, and shown that tumor cells do take up these nanovesicles, and the DNA is delivered in tumor cells. So those are all very important findings, and this delivery system is the basis of our oncology gene therapy program. So I'm just going to touch on the mechanisms of action. Frankly, I'm not a scientist myself, so I can't get too deeply into them. But there are three known delivery mechanisms or mechanisms of action. One is controlling cell signaling. Human cells, or animal cells for that matter, receive signaling from DNA to show the cells how to grow, how long to live, and that kind of thing. If the signaling goes awry, then the cells can proliferate out of control and cause cancer. Similarly, there are apoptotic pathways which lead to natural cell death. That is, our cells are programmed to die at a normal time. If they don't die when they're supposed to, then again, they can proliferate and lead to cancer. And finally, our gene therapy modulates the immune response. It promotes immune activity against cancer. On this screen is data from our first clinical trial of our gene therapy, using our gene therapy as a monotherapy. This trial, done at MD Anderson, enrolled 31 patients. 23 were subject to evaluation. They had to have 2 or more doses to be evaluated. Of the 23, 5 showed a benefit, stable disease or better. 3 of those 5 had tumor responses, that is, reductions in their tumor size. One patient had a disappearance of their tumors by a PET scan, that is, measuring metabolic response. That's the one that you see on the slide here, and you can see that on the left side there are tumors present in the lung and pancreas, and on the right side there's no evidence of those tumors. This particular patient lived for over 7 years after treatment. Granted, it's an outlier, but very encouraging. Next is a presentation of our data from the phase 2 portion of our phase 1/2 clinical trial, combining our gene therapy with the EGFR inhibitor ERLOTINIB. So, as I mentioned, patients who do not have an EGFR mutation don't benefit from drugs like ERLOTINIB or Tagrisso, and normally don't even receive them. Patients who do receive those drugs eventually relapse and no longer benefit from them. While I could spend a lot of time on this slide, there's a lot of data here. I just want to hit on a few points. First of all, in this data, we see patients who were EGFR negative. You wouldn't expect to benefit from erlotinib, but when we combined our gene therapy with erlotinib and treated them, they did benefit. So EGFR negative patients benefited from the combination of our gene therapy with an EGFR inhibitor. So that would suggest that we can treat patients outside of that 10% or so that have the markers for an EGFR inhibitor like erlotinib or Tagrisso. We could expand that 10% number to some larger number. So you can see clinical evidence of that proposition right here. We see patients who were EGFR positive and you would expect would receive a drug like erlotinib or Tagrisso. They did receive those drugs. They became resistant to them. And then after becoming resistant, they received our gene therapy along with them, and they benefited again. They were resensitized. We have several patients in that category. You can also see here that we have patients who benefited from our gene therapy after they had already failed immunotherapies, the anti-PD-1s. So we have patients who had failed chemo plus immunotherapy, and erlotinib, who all benefited from our gene therapy. So it was this data, along with our extensive preclinical data from MD Anderson, that laid the basis for our fast track designation for the combination with the EGFR inhibitor Tagrisso. So we think this is very positive data and compels the studies, the upcoming study with Tagrisso. So researchers at MD Anderson have shown that our gene therapy shows synergy with immunotherapies. They've done multiple mouse studies using this combination. They did one study with syngeneic mice and showed that combining our gene therapy with an immunotherapy, whether it's a PD-1 or a CTLA, showed synergy. That is, the combination was better than either drug alone. And you can see on the right there the slides showing mouse lungs, non-treated and treated. You can get a very visible idea of the effect of this gene therapy on the lungs. The treated lung is much clearer than the non-treated one. And by the way, I should have mentioned, you see the reference to TUSC2, TUSC2 on the screen. TUSC2 is the gene that's the active ingredient in our gene therapy. That gene is carried by our nanoparticle delivery system, and the gene encapsulated into the nanoparticles constitute the drug, which we refer to as GPX001. In the past, we referred to that as ONCOPREX. We now use ONCOPREX to refer to the delivery system. I know that's a bit confusing, but we learned that we wouldn't be able to use ONCOPREX eventually as a trade name, so we reverted to this naming system so that we could have a system of clearer names going forward. So after doing that initial mouse study at MD Anderson, the researchers led by Dr. Jack Roth essentially repeated that research in a humanized mouse model. These are mice that have human immune cells injected, and then the human immune cells are challenged with the drug so that you can see the effect of the drug on actual human immune cells. And they did the combination in humanized mice with TUS2, again, our active ingredient, plus Pembro or Keytruda, and then the combination of TUS2 plus Pembro plus chemotherapy. In both cases, there was substantial synergy, and it was found that the combination was significantly more powerful than either drug alone. The combination of TUS2 plus Pembro plus chemo is important because now of lung cancer patients who come into the clinic, my understanding is that they're typically tested for their genetic makeup. If they have the EGFR marker, they're going to get a drug like Tagrisso, an EGFR inhibitor. If they don't have that, then the next in line would be an immunotherapy like Pembro or Keytruda. If they have the markers for a Pembro or Keytruda, they're going to get that. They may also get chemo at the same time. That is a combination with chemo. Many of them get that. And it's important that the researchers showed that our gene therapy in combination with Pembro and chemo is synergistic because that covers a large percentage of the patients who will get an immunotherapy. So we, again, have the potential to treat a majority of lung cancer patients using our gene therapy in combination with one of these other approved drugs. Now I'll spend a couple of minutes on our diabetes therapy. Again, we licensed this earlier this year. It has the potential to treat both type 1 and type 2 diabetes patients. Diabetes is a very large market, about 34 million patients in the US, or 10% of the US population. So again, very large needs. This therapy could potentially replace the burden of glucose monitoring and insulin replacement therapy. This is all based on research and conclusions by the researchers at the University of Pittsburgh. The therapy holds the potential to possibly even be a cure for diabetes. There's a video on our webpage at genprex.com of Dr. George Giddies from the University of Pittsburgh discussing this therapy, in case anyone would like to take a look at that. To talk briefly about how it works, it's an infusion process. It uses an endoscope and a viral vector to deliver PDX1 and MAFA genes to the pancreas. These genes transform alpha cells in the pancreas into what Dr. Giddies refers to as beta-like cells. They're not exactly identical to natural beta cells, but they're beta-like cells which can produce insulin. And very importantly, they're distinct enough from beta cells to evade the immune system. And so that may be the reason that these cells are not promptly destroyed by the immune system as soon as they're introduced. Preclinical studies showed that this diabetes gene therapy restored normal blood glucose levels for an extended period of time. So there's reason to believe that it might provide extended benefit for diabetes patients. Again, that's preclinical. Research is being done in primates right now, which is being funded by that $2.6 million NIH grant that I mentioned. I'll touch on some of our milestones through early 2021. We are on track to complete the scale up of our manufacturing and to be able to manufacture large-scale batches of our final drug product this year. This will support expansion of our clinical trials. We plan to conduct both clinical trials at multiple trial sites and also eventually commercial scale development. We're doing additional preclinical research for our oncology platform, including new cancer indications and potentially new drug combinations. We're conducting our additional preclinical research on our diabetes program. Clinically, we're continuing to prepare for the 2 upcoming clinical trials, and we're on track to initiate those trials early next year. We're working with Addison Whitney on pursuing brand names for our drugs as well as non-proprietary names. We already have one for GPX-001. And of course, we're always working to increase public awareness of our company and to get our news and our story out there, which is, of course, I'm doing right now. I'm always hesitant to talk about comparables. People sometimes want them. Really, nobody is doing exactly what we do. There aren't any other gene therapies for lung cancer in the works that I'm aware of. Certainly no one else using the TUS2 gene. But I just throw these up there. These are available on our website if you want to see evaluations of other targeted cancer companies and what they're doing. These are a series of companies with a range of valuations. You can see that all of them are drastically higher valued than we are, and this leads us to believe that we're undervalued in the market and that it would be a good entry point into our stock. So just in summary, we're developing 21st century gene therapies. We're addressing large markets. We're combining our drug with top-selling cancer drugs. We have a fast track designation, and we're exploring new cancer indications, new drug combinations, and potential partnerships. That's my presentation for today. Thank you very much. I'll turn it over to Ahu now for any follow-up or questions she may have.
Thank you, Rodney. I'm Ahu Demir, the Noble Research Analyst who covers Genprex. Here are the questions I have selected as well as a couple of my own. So the first question, Rodney, is what is the current status of trial enrollment?
Is the question, what is the current clinical trial enrollment?
Correct.
Okay. We are not enrolling patients in a clinical trial at this time. We're preparing to initiate 2 clinical trials early next year. We're enrolling patients in the phase 1/2 clinical trial combining our gene therapy with ERLOTINIB at the University of Texas MD Anderson Cancer Center. But we received the FDA fast track designation for using the combination of our gene therapy with Tagrisso instead of Tarceva. Tagrisso has taken over the market in the United States for EGFR inhibitors. And so we believed that it would be a better path to focus on the combination with Tagrisso rather than the Tarceva combination. But the FDA allowed us to use that Tarceva data to receive the fast track. So that's the current status.
Okay, the follow-up question is, do you expect a stronger synergistic effect of GPX-001 in combination with either Keytruda or Tagrisso based on preclinical or clinical evidence?
Is the question, which do we think is going to be stronger? Was that the question?
Correct. Yes.
I think it's too early for us to answer that. We haven't done a clinical trial with immunotherapy yet. That's one of the two that's coming up. But let me mention, I think more importantly, they address different patient populations. Patients who have the EGFR marker are going to receive an EGFR inhibitor like Tagrisso, but not an immunotherapy. Those patients typically don't receive an immunotherapy. Likewise, if a patient does not have the EGFR marker, then they may be prescribed an immunotherapy, but they will not be prescribed a drug like Tagrisso. So those are kind of mutually exclusive. So one combination doesn't have to beat the other. They're separate populations, separate and distinct, and we're going after both.
Okay, the next question is, what other indications and markets do you plan to expand with your platform technology?
Well, we haven't announced the next indication that we're going after yet. Preclinical data suggests that the gene therapy should be effective against renal cell carcinoma, breast cancer, including triple-negative breast cancer, soft tissue sarcomas. There are a number of others. They don't all come to mind right now, but there are at least a half dozen that we have preclinical data on. We will be pursuing some of those. We're just not prepared to identify them at this time.
And Rodney, my next question is, what are the value-generating catalysts until trial initiations in the next 12 months?
Well, we intend to initiate both of these trials early next year, possibly as early as the first quarter. Between now and then, as you know, I can only discuss here what's been made public. We will be completing the scale up of our manufacturing. We expect additional preclinical developments in that time frame. We expect to be initiating these two clinical trials, as I've discussed. And before then, we expect to be engaging numerous clinical trial sites and identifying investigators for the trial. We believe some of them will be well-known, highly regarded investigators, along with the prestigious clinical trial sites. All of that will be going on before we initiate those clinical trials. And again, now we're in September, and we think we'll be initiating, first quarter, first half of next year.
Okay, this brings me to my last question. What is your cash runway and burn rate, and when do you think the company will become profitable?
We're not giving a monthly burn rate because our expenditures are, based on, or some of our larger expenditures are based on one-time items or unique items. We are saying that we have cash on hand to run approximately an additional 2 years. When we will become profitable, I can't say at this point. It would be premature to say that. We need to either complete one of these clinical trials or receive an accelerated approval even before that, based on data from one of these trials, before we can commence sales. And I talk about that. It's not unheard of for the FDA to grant an accelerated or early approval based on early data from a trial, based on the company performing a confirmatory trial afterward. And that's very optimistic to think that we might be able to do that, but frankly, I'm hopeful of that. So I can't predict at this time when we'll become profitable.
This is all the time we have today. Thank you very much for helping us get a better understanding of the company's story. We would like to, if you would like to get the independent research I have written on Genprex, go to channelchek.com and type the company name or the ticker GNPX in the search box located in the top left company data section of the site. A recording of today's virtual roadshow will be available on Channelchek. And again, thanks to our guest, Rodney Varner from Genprex. I would also like to thank all of you from the investment community who have watched today, looking for more virtual roadshows on Channelchek brought to you by Noble Capital Markets. Thank you.
Thank you all very much.